Hello, my name is Miguel Rodriguez. I am one of the few collaborators on this specific project, and today I'm going to be presenting wheat spike detection using Mask-RCNN for a high throughput phenotyping system. My mentors include Dr. Augusto Souza and Dr. Yang, and I'm working alongside other collaborators/students. Our project uses Mask-RCNN and faster-RCNN to help make our deep learning algorithms more sophisticated. What I mean by this is using Mask-RCNN and faster-RCNN, it helps the algorithms know what is separating what region, and it creates a region that is defined as a spike. This technology is helpful for researchers because manually they would have to count each and every plant to determine yield usually, and in this case this algorithm essentially gets rid of the need to do it and only requires the input of a photograph. And from photograph, one thing that can be interpreted, or an aspect that can be contrived from it, is a spike, also known as spike recognition. And all of our data was gathered from the Purdue University Controlled Environment Phenotype Facility, which is what is pictured on the screen. The problem that we're trying to help the real world solve is calculating or estimating accurately what the wheat yield is, and it's important that we're able to do this because if we know how much a wheat plant yields, then it helps our farmers or any person in the agricultural side know when they're planting too much. But more specifically for this project, we're trying to solve wheat spike detection, or just trying to get an accurate read of detecting wheat spikes. Right, this is our methodology / our process of what we are doing. We are using Mask-RCNN to separate our images into the attributes that we want to take into account. In this case, it would simply be for mass detection of wheat spikes, and it's really helpful to use mask because when two spikes intersect, normally one would disregard, but since we're using mask, we're able to tell, or hopefully able to tell, that those spikes are two individual instead of being none at all. And we're using faster RCNN to quickly be able to go through that without taking too much time in the sense of running the algorithm. In order to train our algorithm, we had to use some image augmentation. This includes cropping the images to reduce their image size, which helps us be able to contain the images without wasting too much space. But a consequence of this is that we have a limited amount of spikes. What I mean by this is each plant only has a set amount of spikes, and we were given a set amount of plants to work with because all the plants are gathered from the phenotyping facility. So as a result of this, we needed much more data for spikes to be able to train our model. So what we had to do was rescale / rotate, and this can artificially increase our data size without the necessary need to gain more photos. And one of our other steps that we have to use to be able to train our program is labeling, and this wasn't through an algorithm. We had to manually do this. As you can see, essentially we're using an image annotator to create as tight a space as we can around each spike, and we're calling that attribute or labeling that attribute spike, and we do this through the number of images that we had, and through each image each attribute is numbered and it's entitled spike.
My name is Zhengsen Fu. I majored in computer engineering, and I'm a member of the Digital Phenomics group. And I'll introduce you guys to the result and evaluation of our model. As you can see on the right, this is a result generated by our model. And the background of this picture is colored to black and white. The masks are those ones remains colorful. The masks generated by our model are a set of 2D arrays, and its coordinates are mapped to each pixel on the picture. If the computer thinks this pixel belongs to the object, then the corresponding variable in the 2D array would be true, and all the pixels that belongs to the object would be true, and everything else that is background would be false. The center of masks are calculated through k-means, and it is labeled as green dot on the picture. The bounding box are generated through faster RCNN part of the Mask-RCNN, and it's labeled as green rectangles, and its confidence level is labeled on top of the bounding box. The number of pixels within the masks are labeled on the bottom of the bounding box. The width and height are also calculated, but they are not labeled on the picture. They are exported as CSV file along with all other parameters above. Also, as you can see on the previous slide, the recognition on the picture was actually pretty successful. The confidence level was pretty high and it's pretty precise, but on this one, it is not. We evaluated the model with the method of mean average precision, which basically evaluate the area of overlapping with comparing the labeled ones, manually labeled ones, and the bounding box generated through the model. And we can get a pretty high, actually very high mean average precision from if we are using the data from the facility to do the recognition. It could be at as high as 85.151%, but we get nearly 0% mean average precision if we use spike images from, let's say, internet, as the one on the right. It can barely recognize any spikes. This is because the model was trained from the data with the data owing from our facility, which could cause overfitting problems. The model was not perfect, so in the future, we are planning to use the ground data truth to see generated by our evaluation method to see if there's any problem with the model. And in the future, we probably will relabel all the dataset. We probably will have the overlapping spikes in the labeling. And also, we are planning to do kernel counting in the next step. That is to isolate each spikes and then count the kernels on the picture. This can be realized by using another model that are specialized to recognize the kernels, and also this must be done with higher resolution pictures, because we can't see the kernels with the current data set. And with the kernel counting functionality, we're able to estimate the green yield per plant. This would be much faster comparing to manually counting, and also it saves a lot of human resources. Thank you for watching the presentation. If you have any questions or anything you want to discuss with us, please feel free to contact us through these methods.